Yeah.
Okay. So thanks for the kind introduction, and thank you for the opportunity to speak this morning or afternoon, depending on where you are. So good morning or good afternoon. Over here is 4 p.m. in France. So I will talk about some of the science and also a bit about the techniques that we use that I use at ID 21 to do some life and environmental science. It will be more on the environmental part. Um, so, well, I want to start just by saying like what is the importance of using synchrotron light. The main concept is that this radiation is has a high brilliance, which means that you have a high concentration of photons in a very small area. And if you compare these synchrotron sources to the normal X-ray tubes that you use for the techniques in laboratories, as you can see, is several orders of magnitude. So the fact that this light is so brilliant allows us to do very interesting experiments which go faster or have enhanced or much better spatial or spectral resolution depending on the technique. So in terms of the facility where I have been working for the last 15 years, so is the European Synchrotron Radiation Facility, which is located in Grenoble, France. And so is this the only truly internationally funded synchrotron source? So you have several countries contributing to its budget, which it's very interesting financially speaking, but mainly it's very diverse in terms of nationalities of all the scientists that are coming here every day and do their experiments. So I find that this way of operating is really, really fruitful for the science that it is done. And as you can see, there's countries like South Africa, which contribute to the ESRF, and this has advantages for the countries that participate to the budget, that they can come, their users come here, 3 users come here for free to do their experiments. So all the travel expenses and the hotel and meals are paid by ESRF. It's actually the money of the countries. It's in the way they retrieve or recover um, this money they're spending there, funding the ESRF. So we have 42 public beamlines, more than 150 scientists working on all these beamlines and many research visitors. So like all synchrotrons, there's proposals periods, one every 6 months. You can submit proposals. It doesn't matter if you are not belonging to these countries. This is not a criteria. If your science is good, then you get some beam time. The only difference will be that in that case, depending on you will have to pay your expenses to come here. For the rest, the use of the machine and everything is for free, as it is done in other synchrotrons. And so the ESRF is also one of the first 4th-generation sources. After an upgrade that was done in 2019, we got this extremely brilliant source, is the way we call it, or the EVS source. And basically, like in a few words, what it means is that you have now this is the electrons. So the electrons in the storage ring, which are generating the radiation that we use, are then compacted, much more compact in the horizontal direction. So instead of being this very, very big elliptical or rectangular shape, now you have something which is a bit more symmetrical. This means that you have now higher concentration of photons, but also for X-ray, for imaging beamlines, this means that your source smaller. This means that at the end, your beam size will be smaller depending on the optical system that you are using. So this is very interesting for ESRF. This has been operating since 2020. So at the moment of the COVID pandemic, we were just starting the machine when we had to go and work from our homes. But since then, it has been operating, and we are quite satisfied and quite happy with this new source. In particular, for ID21, it has several advantages. So as I said, in synchrotron, at the ID21, I'm sorry, at the SRF, we have 42 public beamlines, and there's other beamlines which belong to countries that are members of the ESRF. So I'm working at ID21, so this will be right here. This is a beamline which is short in the all experimental hall, and our techniques are mainly X-ray fluorescence and X-ray absorption spectroscopy. So basically, out of the several ways in which you can detect or you can do imaging, you can detect the emission of electrons or diffraction, the scattering. So we concentrate on the absorption process, so from the photoelectric effect, and we look at the fluorescence radiation, so X-ray fluorescence. So all our detection is done in this X-ray fluorescence mode, even though we have also detectors to do to get the transmission signal. But our main game is X-ray fluorescence for several interesting reasons, like detection limits, but also the multi-elemental characteristic of this technique. So X-ray fluorescence and X-ray fluorescence spectrometry. So we basically shine X-rays into materials. So these X-rays, if they have a given energy, they can kick out electrons from the valence shells from the shells of the atoms, and then there is a relaxation process where electrons come and fill this vacancy, and then you have emitted photons, and these are characteristic of any of all the different atoms. So you have basically electron binding energies. So for example, iron, you will need to have photons coming, should have more than 7.1 kilo electron, 7.2 kilo electron volt. So they can interact with these K1s electrons. Then they are kicked out. And when the relaxation process takes care, depending on from which energy level, from which orbital they are coming, they will have given energies, and these are tabulated. So we know the emission lines at which energies this should be located, and this is very characteristic of a given atom. So that's why we have this elemental sensitivity, and so we excite several elements at the same time, and then we can detect if we use the correct detector. So detectors for X-ray fluorescence, they are silicon drift diodes mainly what is used these days. So you have X-rays shining. There is a current which is created. So we use an electronic preamplifier and a digital pulse processor. And after all this, you get an histogram. In this histogram, you have different peaks. So you can calibrate these channels on the detector and convert into energy. And based on this energy, then you can attribute the peaks to different elements. And as you can see, elements have overlaps. If you have a sample that contains all elements in the periodic table, then you're maybe going to be into trouble. And that's why it's very important to create or to process your data by a fitting routine. But if you have fewer elements and some of them are more concentrated than the other, then you get along pretty well, and you have this multi-elemental sensitivity, so you detect many elements at the same time, thanks to this X-ray fluorescence spectrum collected by the silicon drift diode detector. And since I'm talking about X-ray imaging, so X-ray fluorescence imaging, of course, you have to somehow produce a small spot, right? So for that, we use some optical device. What we use mainly is Kirkpatrick bias mirrors, and we use these mirrors because they have the highest efficiency and because they're achromatic. So what it means they're achromatic is that they don't depend. So you can send any kind of X-ray energy into the optical system, and you still get the same similar spot size. I mean, it depends a bit on the energy, but they are highly achromatic and very efficient as compared to other systems like compound refractive lenses or Fresnel plates, etc. That they have you lose some intensity because they are based on diffractions effect or refractive. And so these are really you send X-rays at a very shallow angle, and so you have total reflection on their surfaces. So the efficiencies are at the level of 90%. So you lose almost no. There's very small amount of loss of the photons that are going to your optical system. So if you have a device where you can focus your beam, and then you put your sample, you have your SDD detector, and the way it happens, more or less, the experiment is that you put a sample, you raster scan it. So you select which excitation energy you will send. This depends on the beamline. So I will tell you what are the energies we can select at the beamline. Then you will create fluorescence. You will catch the fluorescence spectrum in each one of the pixels of a given sample. And if you then calibrate your spectrum and you attribute these peaks to given elements, you will see, for example, in green we are displaying, sorry, in red we are displaying titanium. In blue we are displaying calcium, and in green we are displaying potassium after this fitting routine. And so you can overlay the elements and you have these nice images. This in this case is a xylem vessel of a plant. And now you know where titanium is distributed in this sample. And you can also display this just as an intensity scale. So the blue is where there's no titanium and the red is where you have the highest concentration of titanium. You can also calibrate this so that it becomes more quantitative and it can give you nanograms per area, so areal densities. Or if you know very well the thickness of the sample and the density, then you can get really, truly quantitative like mass fraction data. Uh, so so once you have these, and if you are interested in getting extra more information about that, you can go for X-ray absorption spectroscopy. In the case X-ray absorption spectroscopy, when you begin exciting an element, then there will be this like increase in the absorption. At the same time, there will be it start to produce fluorescence. So as you change the monochromator, you scan it, you have this step function. But if you have a double crystal monochromator, and usually we use silicium crystals, then you can produce so fine steps that then you have access to these modulations. Instead of having just one step, you have really a modulation of the absorption coefficient. So when you plot it, you will see that these features in the curves will represent oxidation states and geometries. So for example, in here, this cerium acetate carbonate, this is cerium plus 3 versus the cerium plus 4, which is found in oxide, sulfate, etc. So like this, you have these now not only you can detect several elements, but then also you can go with this technique and then understand the chemistry, the electronic structure of the material. And this we can detect in fluorescence, we can detect in transmission, with the advantage that fluorescence is much more sensitive. You can go to ppm or sub ppm levels depending on your type of detector and the flux that you're using. And if you go beyond that, then you can just analyze these oscillations, which is a very difficult signal to collect, but it can be done. And then you're on the X-ups part. But at the ID21, our main work is done on SAINS. And so, as we said, we do these maps, and then we can find the distribution of titanium. Then we can go in the locations. We put the beam at a particular location. We scan the monochromator, and just to give you another example, in the previous one, I show you how you can be sensitive to, in this case, cerium oxidation state. You distinguishing cerium 3 from cerium 4, but even within the different kind of compounds like oxide from hydroxide, etc. And in this case, what we have is titanium oxide. And a titanium oxide, you can have at least two kind of isoforms, which are very well known, anatase and rutile. In anatase, what you have is this octahedra orbit band, and just the fact that you are packing the atoms in a different way, the crystalline structure will have some strong impact in the absorption coefficient. So in here, rutile is this gray here, and you see this peak, which is not present in the anatase. So you can also distinguish different isoforms, even though the chemical formula is the same, TiO2, but they are packed differently in the crystalline structure. So X-ray absorption spectroscopy does not impose to have any things which are crystalline. You can analyze liquids, you can analyze gases, soft matter, etc. So this is also an advantage of X-ray absorption spectroscopy. Uh, So with these two techniques at ID21, as in the title of my presentation I mentioned, we do a lot of life science and environmental studies. And why? Well, because we have spatial resolution, which is submicron, even down to 100 nanometer, thanks to a new microscope that we finished commissioning and now it's operating since 2024. So we can go to 100 nanometer, which allows you to see at the level, at the subcellular level in tissues, you can see the distribution of elements. Because we have the X-ray fluorescence, then you have the elemental specificity. But if you couple this with the science part, you also have access to the chemistry or the ligands and the oxidation state and um, the geometry in which the element is surrounded. So you have more chemical, what we call chemical speciation. And because we have high flux and small beam, because this is a synchrotron technique, you have also high sensitivity. So this means also you can scan faster, right? You can really scan large samples or small samples, but with really high resolution, depends. Um, so the energy range that we cover is from 2 to 11 kilo electron volts. So this is a very good energy region for all studies that concern biological samples. We operate both microscopes on the vacuum. This allows us to have very efficient cryogenic environment, which are dedicated to like really designed with the idea that we are going to do studies on biological samples. Of course, in the new machine, we have everything is much more efficient. Everything is much more to the modern standards. So we have very efficient systems to transfer your sample and take care of it and make sure it never defrosts, for example, when you're doing cryogenic analysis, etc. And of course, because we can produce the XAS, then you get elemental distributions, and also you learn about the chemistry of the element of interest. So the team ID 21, because this beamline is not only bio, so as you can see, we have chemists, environmental chemists, people are interested in ceramics and ancient ceramics, so from cultural heritage domain like Clement. Then me, I'm in life science and environmental science mainly. Ida, whose PhD is more in cultural heritage, working with marine beamline responsible, so he's also a cultural heritage scientist. We have, of course, a very active, very efficient technician at the beamline, and our beamline engineer, Muriel Salome, which is also a very good scientist, not only from the engineering point of view. And then postdoc Alexander Suarez, who is more in catalysis and on data analysis for the science part. And Fabian Letor, who is a PhD, who's working on a project that I will present later. So this is the team. We have a lot of support groups, and so it's a big list of people that I have to mention to and acknowledge about the operation of the beamline and the construction of the new instruments. Like in here, we are installing the new Nano S6M. So if you would like to learn more about the beamline, don't hesitate to just type our names and send us an email. We will be happy to help you to decide if our beamline is adequate for your science. So now I want to go more in detail. When I say the tender X-ray, a tender X-ray beamline, which means it goes from 2 to 11 kilo electron volt, if you are not often doing this X-ray fluorescence technique, maybe it doesn't translate really to what you can do. So that's why we use this periodic table in which all these elements which have a color, green, orange, and yellow, these are elements we can detect. Of course, the K lines, we have more sensitivity than L and M lines. So for the heavy atoms, it depends. We have to understand much in detail the case. But all these transition elements up to gallium, zinc, uh, plus very important elements in biology like phosphorus, sulfur. And if the elements are underlined, this means that we can do XANEs. For the ones which are not underlined, as an example, germanium, arsenic, selenium, these we could detect, but on an L edge, for example, and we cannot do spectroscopy. So I would not really sell my beamline for these elements, even though, okay, if they are highly, highly concentrated, then we can maybe do some work on that. But this allows you to see, like, in terms of elements, what we can do. Of course, if you see the element that you are interested in this periodic table, if it's in the green, then perfect. If it starts going more in those, then we have to check. We are very good on this also, lanthanide, lanthanum, cerium, etc. So okay, send us an email if you think you can do some, and we will confirm to you that that your scientific case is really relevant for our beamline, or I mean that we can actually provide you some data. And so we have this XRF mapping as I show, right? You can have the distribution of several elements, then you move the beam in a given position, and then you can prove the coordination, the chemistry of that element. So as I said, like we work a lot with environmental and biological samples, and then you're confronted with difficult some kind of a challenge, right? Because you need to preserve them, and in the way you would like to preserve them is ideally you would like to have them hydrated as they are in nature, but of course this is almost impossible. Okay, in some cases you can analyze on the native state. There are some beamlets that are equipped with very fast detectors, but if you really want to go in the high resolution, really high resolution analysis, or in some cases, if you for speciation, then it's much better that you somehow freeze the sample in time, and much better way to freeze it than to actually freeze it, right? So cryofixation, it's one protocol that you can follow. So you will do plunge freezing of your sample, then you can do cryosections in the case of our beamline, because we work with 2D analysis. So we do mapping in 2D, we don't have 3D, we don't have tomography. And then you analyze in what is called frozen hydrated state. So this looks like, for example, in here I have a piece of a leaf, which is embedded in this optimal cutting temperature gel. Then you can plunge freeze in a serpentine, doing very manually in here, but now we are just commissioning. We will start commissioning a new machine where we are going to try to improve all this. We have improved a lot, but we are still working on developing protocols. And then we have a microtome. This is at the beamline, so you can do the sections of your samples at the beamline. You have to let's say coordinate the way you will transfer. And this is just a schematic of our old the SXM cryogenic stage. Then nano stage looks differently, but basically you'll have a metallic holder, and then you put some support, you put your section, and then an ultralens film, and you do sandwich, and then and this is all frozen. So in here we are at -50 degrees, and then later we put in nitrogen, and the analyses are done at -170 degrees Celsius, thanks to this cold finger that we have installed in both microscopes. And so to demonstrate, this is the protocol that we follow as up to now. I need to really do again these videos with the new machines we're going to be using, because I think the standards are changing. But as you can see, you put those, you try to freeze them quickly. The videos are not continuing. Yeah. And then this is me doing a section, which is a very delicate process. Then after the section, you have to trap it with this eyelash, and then you place it. And here you can see the section of a leaf in the holder already, and it's ready to be transferred. So when I take this picture, this is the way it is going to be transferred. It should be preserved always in this way. So this is like really under nitrogen, liquid nitrogen, in a liquid nitrogen bath. So like this, we can preserve the elemental distributions of the distribution of the elements in the sample. We are close to the native state, but also we preserve the chemical speciation. And when you cool down the sample, you also, to some extent, mitigate radiation damage, because the dose is quite intense in a synchrotron. So at the beamline, we have a lot of research domains, but the main ones are this cultural heritage, because you have seen there is at least 3 persons from the team who are working in this cultural heritage domain. Then it's me and environment together with my PhD, and also a postdoc who is working there. So earth science, life science, medicine, etc. And as you can see, all these 3 here at least, but even earth science, these are samples that come from environment, and samples that contain, to some extent, some biological sample, and they require this cryogenic preservation. So with that in mind, then I'm going to illustrate a little bit with a few examples what we are able to do. Of course, there are many applications. You can look at the papers from beamline. But let's say if an interesting topic in agriculture is the use of these nitrogen fertilizers, right? That we produce by using this Haber-Bosch process, which requires a ridiculous amount of energy, and it provides ammonia, which you put in the soils. But most of this applied nitrogen is not used by the crops, and it's very expensive process. And nitrogen is very important for all plants. And there are some plants, leguminous plants, that are able to coexist with rhizobia bacteria and produce specific organelles in which you can get nitrogen fixation. So you don't need to input so much nitrogen in the soil. These say human-made fertilizers, they fertilize themselves. They produce nitrogen, and also at the end of their cycle, they leave some nitrogen in the soil. So basically, they create these infected cells called symbiosomes, in which you have enzymes that are capable of converting nitrogen gas into ammonia. And this is like really interesting. It's just like the is the let's say biological version of the Haber-Bosch. And it is for free if you have leguminous plants. And so my colleague from Spain, Manuel González Guerrero, and his team and a lot of people is working on understanding how this process takes place and how this can be transferred maybe to other plants, etc. And you have an important thing is a cofactor, which is a iron sulfide cluster with some molybdenum as well. And so with Manuel, we started investigating just the distribution and the speciation of iron in the nodule. So this is like a classical histology, and this is our sketch, one sketch from a paper from Manuel. And this is what you see with X-rays. Here we're overlaying iron, potassium, calcium. You can see like the very reddish cells are cells which are actively fixing nitrogen, and the ones which are more greenish, it's because they are senescent. So these nodules are divided in different zones, and usually the zone 3 is the fixation, and they are very red. So in here you have a lot of cells which are actively fixing nitrogen. So we were pretty happy to see that in the wild types, this is Medicago truncatula, so alfalfa root nodules, that we can get comparable between the sketch and the histology and what we get with the X-ray vision. So this was a precedent. Now, if you take then a cell, and in here I want to show you how we do this SAINS part, right? You have a cell. This is an infected cell from zone 3, and all these dots in here are regions where we put the beam and then we collect a scan. And then here I'm just showing you how iron 2 to iron 3 and the different ligands, whether you have nitrogen or sulfur or oxygen, can change this absorption spectrum. And so the scans are done very rapidly. Actually, in here I'm putting 0.1 second. Now we are more at the level of 20 milliseconds. So this is 100 milliseconds. So we go even faster. So at each spot we run a scan. We move the position because we don't want to record 2 times in the same spot in order to prevent the radiation damage. And then we collect all this data, and we process it with the software. It's called Orange, and we do some PCA plots, and we are able to average the different spectra which are similar, and then at the end we can do an interpretation of this. So this procedure is quite faster. We can also go and acquire 2D, in which you do an image at each one of these energies, and then you can have a spectrum at the end in each pixel. It all depends also on the stability on the sample and the time that you have, because it can be time consuming. I prefer for this kind of thematic to do this approach where we put a lot of points in different locations and then we compare them. And so in here, I'm showing like you have this nodule here, which looks like that. You see it's very reddish because it contains a lot of iron and like hemoglobin, so like hemoglobin in our blood. Here, this is a section that I'm taking with the video microscope of the SXM. So it's not super clear, but you can see, it helps you to see understand what is the sample and how to do the perfect focus. And then this is the X-ray image, and you can see this is 1 millimeter in one direction. And at the SXM, we can do these images with 3 micron resolution. We are counting 0.1 seconds, so we get in 3 hours this image. But actually, right now in the nano, we can improve the resolution of this image to 1 or even half a micron because we go much faster. And so at the end, our images, they have higher resolution. And in terms of quantity of data, then we are the megapixel. So we have megapixel images. In each one of these points, I want to remind you that we have an X-ray fluorescent spectrum in each pixel and that we have to process it in order to get these elements and to overlay them in the way I'm showing. And in the new machine, because the full system is more efficient to transmit to put the flux into the sample, but also because we have 2 of these 5 element detectors, so we have a larger called solid angle of detection, and so all this combines, and at the end it allows us to get improve quality of the data. So much more data we get much faster. So just scientifically speaking, then if you begin to, so my colleagues, they have some candidates of genes that are considered very important for the transport of iron into the symbiosome, right? And so, for example, ferroportin is one that will transfer iron into this part here close to the bacteroid. And if you block this, then you check distribution of iron in the wild type. You see a lot of iron. These roundish cells, these are the cells that are actively fixing nitrogen, and then you see a decrease of the intensity of iron and also less of these roundish cells. And if we look at the speciation, then there are changes in the speciation as well. So indeed, it's a big difference when you remove this, and like that you can start validating that what are the most important genes. I mean, this is a complementary techniques. There's a lot of many techniques that are also used to explore this thematic, and you can see in this reference. So now another topic, which is the one we're working with the current PhD, Fabien Letor, and it was and now Dr. Blomaer, who was PhD 2 years ago, and it's about cacao and cadmium. So the problem with cadmium is that it's highly toxic. It produces, you can have renal dysfunction, osteoporosis, etc. It's a carcinogen, and we are exposed through our diet, cereals, fish, but now we realize also cacao. Because the cacao plants are able to translocate even if there is low concentrations in the soil, you can get similar concentrations into the bean, and that is an issue. So in particular, South America, and I want to say is not because the locations where they plant are polluted. It's because this is a geological background which is slightly higher, and this causes that the cacao that comes from some countries in South America cannot be imported in Europe because we have new regulations in Europe for dark and milk chocolate. So you need to blend, and so this is a big socioeconomic problem. So there is a toxicological basis for doing this and having these limits, but it has some kind of socioeconomic consequence. And it's very important that we understand how we can diminish the cadmium concentration in the plants. And so for that, you can use X-rays. So we did this experiment at the beamline in here. So what I'm going to mainly show you is data from the branches. So we collect these branches. So my colleagues, Geraldine, Saret, and Esther, they went in a farm in Trinidad and Tobago. They collected the samples under cryogenic conditions, and the frozen samples arrived at the beamline. We did the sections, and in here, what you can see, this is a branch. So I don't have a scale, but this should be about 600 micron in this direction. And so we are close to this part, the cortex, and we observe these kind of crystalline, very roundish structures. And we thought immediately, okay, this may be because they had a high concentration of calcium and strontium. This may be calcium oxalate, which is produced in many plants. And we noticed that the calcium oxalate crystals contain cadmium. And so we probed then the SANES, I'm not showing, but then we demonstrated that in these calcium oxalate crystals, in some of them, not in all of them, you have cadmium, and that it seems like the plant is storing this cadmium in the branches. The branches had the highest concentrations of cadmium. and most of it is stored in these calcium oxalate crystals, which means that there is an important role of these crystals or of the calcium nutrition in cacao that could aggravate or decrease the content of cadmium, because if you would not have these crystals, then more cadmium would likely go into the bin. So this is one result we got right now with the PhD thesis of Fabian. We're looking at different let's say nutrient deficiencies like iron, manganese, zinc, etc. And we want to understand what is the role of all these nutrients in the transport of cadmium, because cadmium is a hitchhiker. It will use transporters from these other elements to go into the plant. There are no specific transporters for cadmium because it doesn't have a biological role. So this is one result, right? Crystals these are involved in cadmium storage in these cacao cultivars. So we are working on that, and we will have more news soon. Then another interesting project is in collaboration with a group in Portugal, Sandra Rodriguez, Diana Diaz, and also my colleague from Toulouse, Astrid Abelan. And in here we are talking about the use of nano-enabled agrochemicals. So here you have what happened is that in vineyards you can use copper sulfate to prevent infection, so it's as a pesticide. And in fact, if you see, you can get the leaves are quite greenish, and when you water them, then you will wash this and it go in the soil. But if you put nano copper oxide, then it's more transparent. It sticks a lot to the leaf, and it has more or less the same pesticide efficiency, so preventing the infection from fungi. So this is one example that I'm not going to present the details on the copper, but basically what we are testing is putting the particles at the surface of the leaf, and then we want to know how it goes into different tissues and how is so its localization. Well, its localization and the chemical transformations across the tissues is what we want to see. And so to illustrate how we do that, I'm going to use an example with zinc oxide, which is a nutrient, important nutrient. So we are more on the fertilizer part. This is more on the pesticide, like the copper story. So this was the thesis of Sandra Rodriguez, and here you have the publication. So we're comparing zinc oxide nanoparticle bare and zinc oxide with a phosphate shell. And so in here you have the comparison. This is a cross section of the leaf, right? Like the one I presented in the video. So here, this is the surface where we put the nanoparticles, and zinc is in red. So where you get this kind of pinkish color. See, if you put nanoparticles, you have zinc going to the epidermis in both sides, the side which was exposed and the non-exposed. But if you put phosphorus, this phosphate shell, then as you can see, it doesn't necessarily stay. It doesn't stay so much as in this case in the epidermis, and we seem to have a more effective mobilization of zinc towards other tissues of the plant, which is very interesting. If you put zinc oxide, then it stays. It goes to the epidermis in the leaf. If you put phosphate, then it moves along to other plant tissues. So this is a very interesting effect. Um, then I want to say that when you're working on nanoparticles, of course, if you want to do SANEs, you have to have very good fixed exit monochromator. So the stability of the beam needs to be able to stay when you scan on the particle of interest. So for example, in here, you have a particle which is about 400 nanometer. And if your beam, as you're scanning, is doing this, you like going in and out of this hotspot, you will get a spectrum which is impossible to interpret. So one of the upgrades of our beamline is to install a new crystal monochromator that actually allows us to stay in nano objects and do SANEs. So here in blue, what you have is the spectrum of a model compound. So the zinc oxide nanoparticle, we put big quantity of it and we don't use a focus beam. And this is what we get out of this 400 nanometer pixel. And as you compare this, we can really say in here, in this spot, we have zinc oxide. So there's still some zinc oxide in the tissue, which is traveling in the tissue layers. And what we do is we collect in many locations along the map that we acquire. So again, in the pinkish part is where you have more zinc. And then this is a PCA plot that we produce with the Orange software, where we compare, for example, fumarate, which is a carboxyl, and phytate, which is a kind of a phosphate bound. And in here we have the original zinc oxide nanoparticle. And as you can see, whatever we find in the tissue, except for a few spots like this, it goes from the carboxyl to the phytate composition here. So you no longer see zinc oxide nanoparticles. This is coming from a reference. So we have a transition from fumarate. So basically, this is what I was telling you. Like we want to understand the chemical transformations of these materials. When you put on the surface, you have zinc oxide. When it starts moving through the layers, it becomes something that goes from the fumarate to phytate. And this depends a lot on the physiology and the way that the plant is managing this extra zinc source. And that would be it. I want to say thanks. Thanks to you for listening and for connecting to this seminar, and thanks to my colleagues Geraldine, Astrid, Sandra, Manuel Gonzalez. Obviously in here I have a long list of colleagues from the ESRF, because a lot of this science is happening thanks to their support in running our equipments and then designing the new equipments as well. So thanks to all of them, and also of course to the team of ID21. And if you have any questions, then I would be more than happy to begin discussing with you and trying to answer whatever doubts you may have. Thank you.
Thank you. Thank you for a very elucidating talk. I saw there were some questions already placed in the chat. So that's from Abedi Kabedi. If you wanted to go ahead and ask some of those questions.
Okay,
or you could go ahead and answer some of them.
The thing is, because I am sharing, so I have to stop sharing so that I can see more clearly the chat actually. Let me see.
Yeah, go ahead.
Okay. I managed to do this. Got it. Okay. So Aveve Kevede, do you do both hexanes on the fine structure? And why is the X-abs hard? Okay, yeah, I mean, the reason why we concentrate mainly on the Saints part, it's because this is a bit what I was saying. XApps requires that you improve a lot the signal to noise in the part where you go into do the Fourier transform for the oscillations for the XApps. So Saints is a more intense signal. You can do a lot of interpretation even when there is some noise in the curves. It's fast. You can do it so fast that you can somehow prevent radiation damage or control it to some extent. But it is possible if you have some robust material, and right now that we have this new DCM, you can even try to do it in some nano object, maybe of the size of the beam. So maybe, I don't know, 200, 300 nanometer. So let's say is for practical reasons, we stay mainly in the collection of the saints, but it needs to be investigated case by case. It's not that we don't have the, let's say, the hardware to do it. It's more like depends on more on your sample, I would say. What is the nature of your sample in terms of concentration and the size of the objects you are interested? But it's possible to do. But it's hard because it's it's these oscillations hereafter. So you have to get a very clean spectrum. Whereas in here, as you can see, you can already do some interpretation, even though this curve is not the cleanest one, let's say, in terms of signal to noise. Was it clear?
Yes, it is clear.
Okay.
The other question I have is, what is the for the environmental samples? What is the configuration of the sample? Is it just powder or film? What kind of form, for the samples that you're using?
So in the case of all these samples I discussed, so in here you have an example. See, these are sections of a leaf. It's a cross section of a leaf. This is a cross section of a stem. This is a cross section of a root. So they are about 20 micron thick, and in terms of size, they can be a few millimeters. But
Nice.
these are thin sections which are frozen, So these are preserved in a frozen hydrated state. But this is some samples, but others. So in the case of the nodule, is the same. So it's more like this, like if you do an histology. But in the for other kind of samples, if you have wafers, we can have soils embedded in resin and then polished. The ceramics, my colleague also embeds the ceramics and then is polishing. I mean, it's an imaging beamline, so a powder just like that, it would be not that interesting. Or something which is very homogeneous, or where the two-dimensional structure is not that interesting, maybe is not the best. But in some cases, also people come with soil samples and they scatter the particles on a tape, and then we map them. It's not probably the best, but it can be done.
Sorry,
So,
Hiram,
yes.
can I pick up on that question and just ask? So this means if we're looking, for instance, on studying organisms that have that are extinct and mapping, for instance, elements in bone and skin as we study evolution and evolutionary process, organisms that might be like 15 to 20 inches, this won't work at this beamline.
No, the limit, the size limit of objects for my beamline is for the scanning X-ray microscope. You have something which is 2.5 centimeter times 2.5 centimeter would be the largest one, because I mean, because the instrument is like that, but also it will be impractical if you want to scan this at 1 micron and you have a 3-inch object. So yeah, the largest sample is 2.5 centimeter times 2.5 centimeter. And on the nano SXM, of course, also a very large sample becomes also very impractical, because if the beam is 120 nanometer or 100 nanometer, then at this sort of resolution, then you cannot scan, let's say, 1 inch. But in terms of the size of the sample, we can introduce, and this is room temperature. I want to also be precise on that. So it's like 2 centimeter, 1.6 times 1 centimeter. So it's a little bit smaller for the nano. So this needs to be considered. And then for the cryo, normally there are sections. So the sections, usually the blocks are maximum, I don't know, 6 millimeter. So I always take a smaller portions of leaf, maybe one root system. In the case of the trees from cacao, of course, they had to be reduced in size. So this is something to really, really consider. That's why it's important that if you think that you can do some analysis at a beamline, the first thing to do is to contact us and say, this is the kind of sample I want to investigate, and we will tell you. Because if we don't have maybe a perfect support, we can develop it if we discuss this in advance. But we will have definitely we have restrictions in terms of the size, which are at that level, right? A few centimeters, let's say, 2 centimeter.
Okay, are there any other questions?
I see one about, do you do operando experiments with catalytic
Yes.
environment? We don't, because we operate in vacuum, then for time being, we don't have any development on any kind of operando analysis. So for example, we did some work on batteries. We do, how to say, like post-mortem. So they are stopped at different charging percentages, and then we probe was it the iron KH. But we don't have any setup right now to do operando. So in terms of sample environment, our specialty is the cryo. And other than that, we don't have anything developed. Like if it's an important project and we see we can somehow fit it into our instruments, we can develop it together. But so far we don't have anything. What is the
There is
configuration?
another one about software. Are you
Yes.
using large to do
Right,
the
but-
analysis?
Yeah, can you probably put in the name of the software? Because
Yeah, so for this, wow. It's nice that I don't have any slides. So it's orange software we're using. Okay, orange software is what we use. We have a workflow integrated at the beamline that is looking at every spectrum which is recorded, and it's being plotted in a PCA space that allows you to compare different samples and even subsamples, because we have the possibility to put a marker on each one of the saints or a tag, and using this metadata we can plot and do this. Then after we use this software, and then we try to create groups of spectra that we average, and then after, what people wants to do is linear combination, and the linear combination, most of the users they're using large. So after this software, which is called Orange, and it's a visual coding platform. You have widgets, you connect them. I wish I could. I don't have any workflow open or anything, but so with Orange, we have this interactive data processing happening as we are collecting the data. With 2 clicks, we get the new spectrum. We get the new spectrum, and we can sort of navigate our experiment. But then at the end of the experiment, we begin working with this, mainly we use PCA, you can use other models, and then you can average points which are close together. So in here, for example, you can produce one for all the green, one for all the blue, one for all the red, and then you can export this data reduced already because you are averaging, and you bring it into large, and large is where we do the linear combination.
Mm.
We would like to develop also the linear combination fitting and all the tools within the same workflow using the Orange platform, but so far we have not succeeded to do this and have everything in this. We would like to offer just one kind of software platform, but so far you still need to use at least with these two. And for the maps, we use a software which is free. Orange as well is free, which is called PyMCA. And so PyMCA takes care of the fitting routine. On each pixel, on each pixel, you have an X-ray fluorescence spectrum, and in PyMCA you create a model. You say which elements, everything, and in each one of the pixels you will do this fitting, and then it will create the maps for individual elements. And this software is PyMCA.
okay. Look, can I ask just a quick question? And that is, have you done any analysis of human tissues, especially human tissue that may have been subjected to mercury poisoning? Is mercury detectable with your
Yeah,
beamline?
mercury is not detectable at our beamline. Mercury will be one of those that we work on M-edge, and it has overlap with sulfur, so then it puts a lot of restriction. So in biological samples, it will be extremely complicated to do analysis with mercury. But human samples, yes, we have worked a lot on Alzheimer and neurodegenerative disease tissues that come from tissue banks. Of course, anything which is human origin requires a special permit, and in most cases, we won't be able to do in cryogenic conditions, like the truly cryogenic conditions. We will be forced to do some chemical fixation before, because you have to demonstrate that the sample is innocuous, that there is no virus or pathogen active in the sample, and that there is no risk of during the manipulation. So, but yes, I've done work on human tissue, and also we do a lot on cell lines. Also, if they are of human origin or they are of cancerous cell lines, then we need some permits. So this is also why it's very important to contact us to explain what is the nature of your experiment and the kind of samples and what are your objectives, and we can quickly tell you, yes, your work is feasible. Otherwise, you will spend time writing a proposal and maybe get rejected on the first filter, which is indeed the feasibility. So we could do the feasibility for you before you submit. And help you in other cases. We help a lot of users before they even come to the beamline, so during the submission process and preparing before coming to the beamline and all that. It's a quite intense work, but we prefer to do that and make sure that that the experiments are successful.
Yeah, understood. Okay, any other questions for the speaker?
One more question of something general.
Mm-hmm.
The motivation of this meeting is to make sure that people, particularly in developing countries, particularly Africa, learn about these resources. The hope is also decision makers know the value of this resource and its value in education and environmental studies. It would be for this type of talks and future talks, it would be very helpful if the speakers can add a map or something to see the user distribution. For example, in this important work, I'm just wondering how many countries from Africa, for example, are participating, contributing to this research. I believe you mentioned South Africa is playing
that
a
is
big,
one thing that maybe I could take the opportunity to to advertise. I know that the South African user community, because there is a scientist now here who spent good amount of time working in South Africa. They come a lot to the tomography beamlines, but at ID21, I think I've had maybe 10 years ago or 12 when I was I was in my postdoc. I had 2 users that came and joined a group here from Europe, from South Africa, for an experiment. And other than that, to my knowledge, I don't think we have any other active users at ID21. I'm not saying at the SRF level, I don't know. So it's definitely something to consider. And in this particular research examples I described, it's mainly groups from Europe, and I have other projects which involve colleagues from Mexico and from Brazil. But yeah, it could be nice to have more and more users from other countries, of course.
Yeah, just another additional point to make is, remember, it would be very nice for African students to travel to Europe, do their research back and forth. That would be really.
Sorry, it's cutting somehow. Hello?
Okay.
Yeah.
Yeah, I stopped because somebody's talking. So it would be very interesting. Remember, difficult times are coming, and traveling will be still difficult. But what would be nice for this community to do is to help African students and researchers to have access to the data itself. If they don't need to be there physically, but they can do data analysis for their PhD and master's work. Perhaps if we can talk about how data can be accessible and how, for example, they can collaborate with you in things like that.
Yeah, well, after and during and after the COVID crisis, we were forced to implement ways in which users can interact with the machine and the control of the beamline remotely. So remote users, even though is not as easy to do as to logistically speaking, it can be complicated, and it puts some stress in the local contact because obviously changing sample and all these relies then in the person who is at the beamline. But this is possible. So you can, if you cannot do the travel, things can be arranged in a way that you can still do your experiment and control the beamline remotely. That's one thing that you're right. If traveling is complicated or too expensive, this can be done. For example, we have one example I have is with some colleagues and some collaborators from Australia. They have never been at the beamline, and we have done something like 3 beam times with them, and some papers also published. So that's possible. And then in terms of the data, well, the data is all recorded in here, but you download through a portal. And in our case, for the beamline, we have a lot of the data processing is done using clusters and computing resources of the ESRF. But after the data is reduced, it's not so heavy that it can be, let's say, maps and scenes and all this can be like the final processing can be done in a normal terminal. So you can copy the reduced data and work on your own institute, which is actually we try to go as much as we can to a point where the data has been already fitted, maps are produced, and then the user needs just to go and see the different elements and understand their correlations and produce the nice figures for the papers. And for the scenes is the same. If we produce 1,000 scans, we will try to give them a database that can connect in this orange software where they can already know where they're going to be doing their averages and perform, or already they produce their averages before they leave. And then after, it's less data they need to interact with. So it's possible to do that. And also it's possible probably to consider about some kind of a collaboration, in the sense that some groups may have maybe lagging in the processing of some data and maybe are interested to ask a master student or someone to work on the data process for them. This I don't know a lot of examples, but
So
usually
this Hiram, and I think it's Adibe. There's a comment, a follow-up comment from Mac in the chat that the main barrier is funding for most of the African universities. And I'm going to throw in, for those of us in regions like the Caribbean also, where many people don't even know what a synchrotron is. It can be useful, Mac says, to communicate on different possibilities to apply for. I'm gonna mention before you come in, Hiram, and I guess most people know about the LAMP program, where the fast team program, where you can spend faculty and student, I think it's 2 months, and my student and I, who are possibly, and Mac is saying the AfLS project should reduce this gap. There's still a big gap because the LAMP program is limited in the numbers it can fund. But one of the things following, I want you to respond to Mac's comment, Hiram, and I also want to throw in that, you know, when we went, my student and I, some years ago on the LAMP program, we came back with a lot of data, and as you were saying, you know, the time was short, and this is all very new, and processing all of this data, we still are not there. This is peer distribution function, which is also very new to us. And so that line of communication when you are visited to continue data processing, And you know, even access to software for data processing must be
Yeah.
included in this. I'm gonna stop there so you can comment on Mac's comment here.
Yeah, of course, this funding barrier, of course, is what complicates for countries that don't have sources which are close, and that the trips are long and expensive. It's of course something which is not making synchrotron a more popular, synchrotron techniques more popular, let's say, and more how to say, more the democrat. But yeah, I mean, programs like LAMP are useful. Then also, I think funding mechanisms or joint synchrotron facilities like this, like the European synchrotron, are maybe a way to, because this is what happens, right? If you become associate country or member of ESRF, then 3 of the scientists from that country come for free to the, I mean, let's say for free to them. No, the government is obviously putting some money in there, so it's in the way that you give back the money. And then also, once you become part of the consortium of the ESRF funding countries, then a lot of effort is done so that you get training and that you get opportunities to visit, and that some of us will go sometimes to the countries or do webinars like these to try to train the users and to boost the user community. So that's another, and probably the countries. This is what I was thinking for, that could in Mexico could be useful, is like if you have some kind of an association or something like this where you can actually allocate some money from a funding source that could be available for users in a more rapid way, because when you apply for beam time, you don't know you're going to get the beam time, and you don't know when you're going to get the beam time exactly. So it would be nice to say, now I have a proposal, it's accepted, my experiment will be in that month, and then in that moment you can apply for these funds, right? Because otherwise, if you have to put it in the context of a big, long project, then this funding, maybe if the group is not successful to get beam time, then what do you do with this money? And if it needs to be like that, then it's also only groups who are going for these big grants that are getting the access. So I think there should be some mechanism that says, okay, if you already made a proposal, it has been peer reviewed, because all synchrotrons is peer reviewed, the proposals. Somebody has said that your experiment is interesting and there's possibility for you to do this beam time session. And at the end, I mean, okay, it's expensive for the group, but at the, let's say, governmental level, is not even that expensive, to give the funding for the trips. Because at the end, when you come in here, the machine, you use it for free. The computing resources you use for free. The computing resources are available for you for 3 months, but you can even get more time if you need. So at the end, everything, once your experiment is approved, at least at DSRF, there's no charges for other things. Cryogenic experiments, we consume liters and liters of nitrogen. This is not transferred to the user. And it's irrelevant if you are from the countries which are funding the ESRF, or if you come from another country like US or Mexico, or some place in South Africa or Nigeria or any other country. Is not. So it's mainly the funding limitation is specifically for travel, I would say. And so maybe there should be a way to organize this. The LAMP program I've participated a few times. I think it's a very interesting tool, because it's a 2-month stay, and in 2 months you can learn a lot of things. If you're new to the synchrotron, if you're new to a given technique or a given beamline. But it's true that maybe at the end of the 2 months, you're not completely autonomous, but you would be even less if you never come, right?
Okay, there's any other questions? We've come quarter past the hour. But if you have any questions, you can actually always contact the organizers at the Caribbean Light Source or the African Light Source, and we can pass your questions on to the speaker, who actually has a public website, so you can actually reach him directly. So if there's no other questions, we'll close the session today. Thank you very much for attending.
Thanks a lot. Have a nice day.
Thank you
Thanks.
very much.
Bye.
Bye, everyone.